So now we have our industry talk. It's Ieva Drulyte, who is going to tell us about cryo-EM. I'm sorry, I probably butchered that.
It was really good, thanks. Okay. So hello everyone. It's really nice to be here, and thank you for the opportunity to present today. Please bear with me. I'm going to have 2 clicker pointers, not 1. And also, thanks very much for the shout out, Pavel. Those tomograms are really well tracked, so went well. Great. So just a few words about me. I grew up in Lithuania, and then I moved to the UK to do my undergraduate at York, and then I really got sort of inspired by all the work in the York Biochemistry department and sort of entomology and structural biology, so I thought I need to learn more about that, and then that inspired me to do a PhD at the University of Leeds at the Asbury Centre, and then this was sort of coincided with the whole resolution revolution. So I was really there from the beginning, and I just really loved the technology and wanted to know even more. So that brought me to my next step in my career, and I joined Thermo Fisher in 2018 as an application scientist. Yeah. So I know that pretty much every talk is using cryo-EM here, and there are also a lot of people in the audience who've been doing this for a very long time. But I also know that some of you are quite new to the field, so I thought I would start a little bit earlier on and just start with the fact that there are 3 major branches of a cryo-electron microscopy. So we have single particle analysis, cryo-ET, and also large volume analysis. And I will cover all of these a little bit today. So if we just start the beginning of the resolution revolution, we had this was the advent of direct electron detectors alongside some other innovations in hardware and software, and that led us to go from blobology to high-resolution structures. And we also saw cryo-EM being named the Nature Method of the Year 2015. Of course, the next big milestone in the cryo-EM development was the Nobel Prize awarded to the 3 pioneers of the technique that everybody is very familiar with. And then if we fast forward again by a few years, we had another really important milestone to us is finally achieving atom resolution in the protein structure. And I think this really shows that all of these milestones are happening in a sort of fairly quick succession, and we still have so many things coming out in this technology, and that really shows that cryo-EM is still in this growth phase. It has not matured the same way that X-ray crystallography, and I think if you look at the PDB depositions, it is also really clear that we are really still in this growth phase. And that's and you can see why Richard Henderson has famously said before that by 2024, cryo-EM will overtake X-ray crystallography in these depositions. And I don't need to tell all of you that it has been used for studying viruses for a really long time. If we look back all the way to the 50s, we were using negative stain to look at samples like TMV, adenovirus, and then, of course, these 2 structures here are the first structures by cryo-EM. And it's also really not surprising to anyone that cryo-EM was used a lot in the really early stages of the pandemic. So I think it's quite astonishing by how quickly there was the first structure of the full spike, the cryo-EM structure of the RBD with this receptor, and as well the first tomography data that came out at the beginning of the pandemic. This was just sort of at lightning speed. And we also had a look at some of the statistics at the time. So just in the 2020 alone, there were 80 publications studying SARS-CoV-2 using cryo-EM. And also, coronavirus spike protein could have its own field devoted to it right now, because there are already over 1,600 depositions at the MDV of just this protein. So that was just a little introduction to EM and how it relates to virology, at least in my eyes. And now I would like to use the next couple of slides to talk a little bit about how we see the future of single particle analysis before I go into the main topic of the talk. So the way we envision single particle analysis that we have to really work on automation and high throughput. We have to move away from this sort of paradigm of one microscope, one user, one sample per 24-hour session, because, yeah. So let me tell you a little bit more about that. And I want to use one example. So I want to use a Glacios microscope. When I joined Thermo Fisher in 2018, we had the very first Glacios that was ever manufactured installed in the nanoport, and we're trying to collect data on various different samples, collecting at 25 images per hour, perhaps 30 at the tops if we really push some things, and we're getting good structures. But if you just take next 5 years, if you fast forward 5 years with some software innovations such as AFIS and also hardware changes like two next generation of detectors leading to Falcon 4i and fringe illumination, now we can routinely achieve 650 images per hour on a Glacios 2. I've even done an experiment where at certain imaging parameters it went over 1,000. So it's really a huge increase in throughput just from combining several different innovations into one place. And another thing that has happened really recently is that we are trying to, like the rest of the world, go smart and to add more AI and machine learning into SPA. So we have launched this new smart EPU. For those who don't know, the ecosystem consists of 3 different parts. So the data acquisition software that everybody is familiar with, including EPU multigrid. But then on top of that, we have this module that you may have if you want to do EPU quality monitor or embedded cryo Spark Live, and that will monitor all data collection on the fly. And then that information can be used to actually, for feedback loops, to adjust data collection parameters. So it sounds really simple, but actually it would save a lot of time if people did not have to really monitor data collection. So computer can decide that that grid square is not worth collecting on, or something happened to the defocus values I need to adjust. So for example, one of the plugins in this ecosystem is smart hole selection. So I'm sure a lot of people in the audience have had the experience to add 20 or 50 grid squares and then manually add the holes they'd like to collect from. So that really takes a lot of valuable time. To really help with that, we trained this neural network to identify good and bad holes. So right now, in this ecosystem, you can click one button, and then it will select the holes that it deems to be good, like here. So avoids cracks, empty holes, or big contaminations. Another nice example for automation is this EPU multigrid for screening. So, at least in my PhD days, I would pick 3 or 4 grid squares in the grid and then navigate to those grid squares and try to take 3 or 4 images in the grid square to make an informed opinion. So now you can actually just tell multigrid to do that for you. So you say that you want to take 5 random images. They get distributed across the ice gradient from as many grid squares as you want. Then you can add all grids in the autoloader in the queue with just 2 clicks, and then you say, run it, go away for a few hours, and then just view all the data in Discovery Viewer. So that's just on your browser, as long as you're in the same network as the microscope. So that should also save a lot of time. And now I would like to show a little bit of a result as well. So when we saw this huge, with the Falcon 4i, when we saw this huge increase in the throughput, we really wanted to ask ourselves, is it possible to really go away from this paradigm of 1 structure per 1 microscope day? So can we actually solve 12 structures in a single day on the microscope? So we initiated this project at the really early stages of the pandemic. So of course, it made really total sense to focus on SARS-CoV-2 epitope mapping. So for this work, we collaborated with 2 places. So with Utrecht University, who are experts in coronavirus biology and really know how to make really good quality SARS-CoV-2 spike, and also with Takeda Pharmaceuticals in San Diego, who provided us with 12 monoclonal antibody Fab fragments, all from preclinical development, all from sort of taken from different sort of ways of production. So we combined the 2, and in a grid, and then we made just 2 grids per sample and screened them on the glaciers, just 50 images per grid. Takes about 5 minutes just to check that the particles were there and that they were classifying into 2D glasses. And then we took the 12 best grids, that's how many fits in the autoloader, into the cryos in the nanoport in Eindhoven and collected 1,000 images per grid. So with today's speed, that's 2 hours. 2 years ago, it took us 4 hours, but today it will be 2 hours per sample. And then we processed all the data on CryoSpark. So we were really, at the time, all the papers out there would use 6,000, 7,000, 8,000 images for SARS-CoV-2 with a receptor or a Fab. So we were fully expecting that half of them would not work. But we thought we'd have something. But actually, we're really pleasantly surprised that all 12 resulted in sub-3 angstrom reconstructions. So you can see here globally refined maps, and you can already see that some of the RBDs, only 2 are up, some are 3 up, and they're closely together, some are spread out. So these are all unique maps from each of the Fab. Then we've performed local refinement for all of those global maps. So here in this little video, you can see sort of the quality of the density in the RBD and Fab region, which really allowed us to accurately build both the RBD and the Fab, and as well the S2 region was really well resolved anyways, but also that is shown. So we published this as a white paper, and so the academic leader of this project is Dan Hurdes. He was chairing the session earlier today, so please feel free to talk to him more about this work. And now in the next slide, I can show the final result. So here are the 12 refinements, local refinements, with RBD colored in blue and the Fabs colored in yellow and purple. So you can see that all these Fabs are occupying really different epitope space. It's quite varied epitope space, so we're able to accurately define all the epitopes and classify all the Fabs. And yeah, so the resolutions we achieved were about 3 to 4 angstroms. Great. So that brings me to the second part of the talk. What is next? And any ideas in the audience?
3 microscopes.
Yeah, well, we think it's cryo-electron tomography. So if we look at the trends again, I love these graphs when I see another talk, so that's why you see so many in this one as well, I'm sorry. But if we look at the trends, yearly depositions from 21 to 22 nearly doubled cryo-ET depositions to electron microscopy database. And another nice trend is if you compare what cryo-ET, what kind of trend is following to that one of SPA, it's about 8 years apart. So I do really believe that this is hard to ignore, and I think it's really picking up at fast speed, and I think we will see a lot of really interesting stuff to come out in the upcoming years. So cryo-electron tomography, unlike SPA, it's a really versatile technique in terms of what kind of samples you can look at. So you can go all the way from purified proteins or purified viruses just suspended in buffer, all the way to cells and tissues. So I would like to touch all 3 of these branches a little bit in the talk today, and I will start with proteins and viruses. So here you see one application result from Eindhoven. So we looked together with collaborators at this equine and infectious anemia virus. You can see that the particle is not symmetrically round, so you cannot use SPA to look at its structure. So for that, we performed subtomogram averaging, and now you can see a zoom in into this C6 hexamer of the capsid, and in a second you will see a monomer from this reconstruction that has 2.9 angstrom resolution, but in fact, some parts of the map go much higher, all the way to 2.4. Another example from a little bit earlier on, it's this HIV precursor structural protein GAG. It forms these assemblies in vitro, and for a long time, it was only hypothesized that it was forming this sort of druggable pockets, and so we used cryo-ET to actually visualize this pocket for the first time, and to about 4 angstrom resolution. So now I showed you 2 examples that are around 3 or 4, and a lot of the criticism always comes after the resolution you can achieve for cryo-ET. So we wanted to do a really, really fun experiment and see what the technology today, the hardware and the processing software is capable of if you sort of take away the limitations from the sample. And there's only one sample that you can use that will take away all those limitations. So we did cryo-electron tomography on apoferritin grids. Yes. And to our surprise as well, just less than 2 hours of data collection time already went to sub 2 angstroms for apoferritin. And when we processed the entire data set, which was about 100 tilt series, so that takes overnight to collect these days, that went to 1.5 angstroms. So whilst we don't need another apoferritin reconstruction, I think this was a really neat experiment to say that if you are, well, particularly if you're using our hardware and the processing solutions that are out there, those will not be the limiting steps in your experiment. It will most likely be the sample. And of course, this data set is available at Empire, so if you would like to practice processing or practice some other things, you can download it yourselves. So now that brings me to more complex systems of cells and tissues. And as Pranav said, we need to make a window inside the cell. So we need to use technology that is called focused ion beam milling to make a lamellae. So because most of these samples are just too thick for tomography, unless you maybe look at the ends of the cells. But in all of the cases, they're too thick, so we want to mill them to produce these thin slices, and that really will allow us to visualize the contents of inside the cell. And one note about vitrification, so if it's up to 10 micrometers, plunge freezing should be possible. Above that, you need high pressure freezing. Oh, very. Sorry. Great. So I have a really lovely video of inside a small dual beam. Let's take a look at how this process works. So we have this cryo stage, and we can insert 2 grids at a time inside a dual beam into this big chamber. The first thing that happens, we need to coat the cells or material with platinum as a protective layer. Then the whole stage rotates, and now we'll see it tilting, and we will be using scanning electron microscope beam to visualize the surface of the sample and focused ion beam to mill the material. Here you can see sort of representation of a cell, which is very conveniently placed in the middle of a grid square. That's actually harder than it looks. And we make this pattern, and then material is milled away. At first, it's milled sort of fairly fast. It's called like bulk milling, and then you have to polish it. The polishing step is more often like less automated than the first step, but everything is automated these days, but quite a lot of people still polish by hand. Then you can see this green. That's the platinum left over from the top or surface of the cell. So now we will rotate it again, and now another deposition will happen of inorganic platinum, really thin layer, and that's to really protect the lamellae and prevent charging from happening inside the microscope. So once that is done, we can now transfer this lamellae or lamella into the TEM. So inside the TEM, and I think the rest is really clear. But we tilt the stage at increments and take a single image from each tilt, and all of those are then a line from motion, CTF, etc., and reconstruct it to produce a volume, and then you can segment the volume to produce really lovely images of the architecture, or you can perform stop tomogram averaging, or both. So these are the three small dual beam systems that we have. So there's Aquilus 2, that's the workhorse of the system. So we recently celebrated a little milestone of shipping 100th of it in the world. So there's quite a few of them, and most of the publications in cryo-ET will have used this one. Hydra is a really versatile system, and it's also a first plasma FIB for life sciences. And then we also have a new kid on the block, Arctis. So what's special about Arctis? So it is really geared for automation and connectivity. As part of this mission, so it has all three beams, so light microscope for fluorescence, PFIB and SEM, coinciding on the same area on the sample. It can also load 12 grids at the same time. The chamber is also really small, so you don't need to bake it out as often as for the other drill beams. Maybe, I think once a week is what we recommend. So here's one example of 47 lamellae produced in 48 hours on the same grid. And here we can see some zoomed in versions of one of these lamellae. So you can see a search image of a lamellae. You can see really nice thylakoids from the reconstruction, from a tomogram taken from a lamellae. And you can already see that there's not so much curtaining happening, and the contrast is pretty good. You can already see the features in the lamellae from this image, so you can place the areas. In certain situations, that's all you need to place the areas for tomography data acquisition. We also measured the thickness of this particular lamella, and that was 55 nanometers. And we also measured the thickness of 100 tomograms and got an average of 128 nanometers. And here's one collaborative project that I would like to highlight. So we would like to look into Chlamydomonas proteome using high throughput visual proteomics. So Chlamydomonas cell has about 14,000 proteins. So there's a lot going on, and we wanted to see how many of them we can identify and solve structures of. So we collected over 1,500 cell series from about 150 lamellae, with more collections still ongoing. And from this work, we identified so far 20 different proteins and solved 4 different structures. In this slide, you can see some of the examples from these data sets. So we have rubisco particles. They're really tightly packed inside a pyrenoid. Here we see the Golgi apparatus, and over here, thylakoids as well. And here there are 2 examples from mitochondria. So all these inner membranes decorated with ATP synthase particles, also respiratory complexes, and finally, we wait for the right slice. You can see several nuclear pore complexes appearing. And in the next slide, I will show just one reconstruction from this work. So, this doesn't want to. Okay. So we took one lamella, in total 6 TIL series, and reconstructed 6 tomograms, and from that we calculated this 6.2 angstrom reconstruction of a ribosome. But in the wider context of this work, we actually identified 14 different states of the ribosome from these data sets. And that is actually a few more than some recent work that identified 12 in vitro assembled ribosomes. So, of course, in our opinion, it's really exciting, because once you really start looking at these proteins in vivo and you can actually quantify them, you can then ask questions like which ribosome is doing what, where, when, and why. Yeah, it's okay. So last section is on Hydra. It's also a plasma amphib like Arctis, but it has so it doesn't. It besides making lamellas, it can also do some really other techniques such as cryo lift out and also slice and view. So slice and view is one application of volume EM. Volume EM has actually been named one of 7 technologies to watch by Nature, by Nature Publishing Group in 2023. So essentially, this technique aims to, instead of looking into just one section where you may be playing sort of a game of statistics, look at every single section and visualize the entire volume of your sample or a tissue. So the main techniques are slice and view, spin milling, volume scope, and area tomography. And I have 2 examples of slice and view. So one of them is this chlamydomonas cell. So here, 335 slices, each at 6.7 micrometer thickness that were taken, and then they were used to reconstruct this entire view. And I wanted to just highlight the fact that it's just plunge frozen, and there are no stains or fixatives added. So I think you can, I hope, appreciate there's a lot of detail in there. And you can really visualize a lot of different compartments and cellular features. I'm sorry. It's these videos. This one is of a tissue. So that's a mouse brain tissue, and here 129 slices were taken. So I think this is also really exciting because you can actually, retrieve tissue samples from native source, and if you can just use high pressure freezing and then use such a technique as slice and view, you can visualize the effects of, for example, drugs or like disease states and so on. And then last couple of slides for those who are doing tomography and using our software. So the team has been working really hard over the last couple of years to bring a lot of features into this program, so I wanted to highlight a couple of them. We have, a while ago, already added this feature where you can click and add a new tomogram with just a single click, and you can also view all batch positions in this interactive map, and you can, of course, do dosimetric tilt scheme. More recently, we also added lamella angle wizard and also search map alignment to produce these nice search maps, and integration with TomoLive. And finally, we went from sort of a method where you can collect one tilt series per one position into collecting multiple tilt series on the tilt axis. And now it's actually called off-axis or multi-shot because we can also add tilt positions off the tilt axis. So that really drastically increases the throughput, where you can collect a tilt series in less than 8 minutes, and that really allows you to collect over 100 overnight or over 500 over the weekend. So, of course, we also think that it is really important for us to have some sort of way to see whether these tomograms are any good, and in a way that's a bit more automated than somebody reconstructing them by hand. So we actually built and now launched the software called TomoLive. So TomoLive runs on the Falcon server, and it, on the fly, does motion correction in your movies of a single tilt, alignment of a tilt series, and then reconstruction. So just a few minutes after the tilt series is finished, you can actually see the final reconstruction on the web browser. It looks like this. And then there is a bunch of metrics that you can filter. And essentially, it's a really great tool for curation of your data. So instead of reconstructing 500 and figuring out which ones you want to work on, you can just select the ones that you really want. And then what's also quite nice is that you don't need to optimize any parameters whatsoever. It's completely automated, and it just happens in the background. So that brings me to the final slide. Thanks to all the collaborators. Thank you to the organizers and all of you for being here, listening to the talk. And I would like to say that if you would like to know anything more about the products or book a demo, please come and see Ati over there and myself. So thanks very much.
We're a little bit over, so maybe just one question. And remember, the trainees get prizes if they ask questions, so maybe we could have a question from a trainee. Be brave. There's one back there.
So how come, this is kind of maybe subjective. How come you didn't show us a cross-section of like a virus or something?
Cross-section of a virus. Oh, in the talk, yeah, so honest question, honest answer, yeah. In our facility in Andover, we cannot really handle virus samples. So, yeah.
So let's go ahead and have coffee break, and you can catch. Sorry. Really?
I was wondering how are you progressing in automatic feature identification for segmentation, and how you can identify specific objects within your, because you get a lot of interesting images, but to know what is what, how is software progressing?
Yeah, let's take it over coffee, but I can tell you a bit more about Amira. Are you using Amira? Yeah, so in our Amira team are actually working on using AI/ML to identify features and make segmentation much more easier. Yeah, so yeah, we can chat about it more.
All right, sorry for a shorter coffee break. Back in 20 minutes. Thank you.